Imagine a world where no patient ever dies because there was no blood available. No delays in emergency rooms, no desperate calls for rare blood types, no shortages during disasters. That future may sound like science fiction, but researchers have been working for decades to make it real through the development of artificial blood. First, let's understand something important. Artificial blood does not mean a complete replacement for everything real blood does. Human blood is incredibly complex. It carries oxygen, removes carbon dioxide, fights infections, helps wounds clot, transports nutrients, balances temperature, and much more. Creating an exact duplicate is extremely difficult. What scientists are actually developing are blood substitutes. These are specially designed products that focus mainly on one life-saving job, delivering oxygen to tissues when real blood is not available. Oxygen delivery is critical because without oxygen, organs begin to fail within minutes. So if doctors can temporarily restore oxygen flow, they can buy precious time to save a patient's life. The idea of replacing blood is not new. For centuries, people experimented with strange substances like milk, beer, and even animal blood in desperate attempts to save patients. Most of these early efforts failed because doctors did not yet understand how blood circulates in the body. Real scientific progress began after the discovery that blood continuously flows through vessels rather than being consumed by organs. That discovery laid the foundation for modern transfusion medicine. Over time, scientists tried saline solutions to restore blood volume. These fluids helped maintain blood pressure but could not carry oxygen. Later, researchers isolated hemoglobin, the protein inside red blood cells that binds oxygen. This led to the realization that if hemoglobin could be safely delivered outside red blood cells, it might work as an oxygen carrier. Today, there are two major approaches to artificial blood. The first approach uses substances called perfluorocarbons, often abbreviated as PFCs. These are completely synthetic compounds. What makes them special is their ability to dissolve large amounts of oxygen. Instead of using red blood cells, PFC-based products carry oxygen simply by dissolving it in liquid form. Because they are synthetic, there is no risk of transmitting blood-borne infections. They are also not limited by blood type compatibility, which means they could potentially be given to anyone. However, PFCs are not perfect. They must be mixed into special emulsions to circulate in the bloodstream. Their oxygen-carrying capacity is helpful, but not as efficient as natural red blood cells. And they do not perform any of the other roles of blood, such as clotting or immune defense. Their function is limited strictly to oxygen delivery. The second major approach focuses on hemoglobin-based oxygen carriers. These products use hemoglobin itself, either purified from donated blood or produced through biotechnology. In some cases, scientists use genetically engineered bacteria to manufacture hemoglobin. The goal is to create a stable version that can circulate safely in the bloodstream without being inside a red blood cell. This method has advantages. Hemoglobin naturally carries oxygen in the body, so it makes sense to use it directly. Hemoglobin-based substitutes can transport significant amounts of oxygen and may perform more similarly to real blood compared to PFC products. But challenges remain. Free hemoglobin outside red blood cells can break down quickly and may cause side effects if not carefully modified. Researchers must chemically stabilize it so that it does not damage blood vessels or get filtered too rapidly by the kidneys. Even then, most artificial oxygen carriers last only hours to a few days in the body, while natural red blood cells can survive for weeks. Safety is the biggest challenge in developing artificial blood. Any product injected directly into the bloodstream must be completely compatible, non-toxic, and free from harmful immune reactions. It must not raise blood pressure dangerously, it must not cause inflammation, and it must work reliably in critically ill patients. Because of these strict standards, many products that looked promising in early testing did not make it through advanced clinical trials. Despite these obstacles, research continues around the world. The potential benefits are enormous. In emergency medicine, artificial blood could be stored in ambulances and used immediately at accident scenes. Doctors would not need to wait for blood typing and cross-matching before starting oxygen support. In rural or remote areas where blood banks are limited, artificial blood could bridge the gap until proper transfusions are available. In developing countries, blood shortages are a constant problem. Storage requires refrigeration, careful screening, and a reliable donation system. Artificial oxygen carriers, especially those that are shelf-stable, could dramatically improve survival rates in trauma and surgery. They could also be vital in military settings and disaster response situations where large numbers of injured patients need immediate treatment. A universal oxygen carrier that works for all blood types could simplify logistics and save lives on a massive scale. There is also economic interest in this field. A safe and effective artificial blood product would represent a major medical breakthrough and a significant healthcare market. But more importantly, it would represent a shift in how we manage trauma, surgery, and critical care. Artificial blood is not yet a complete replacement for donated blood. It does not fight infections. It does not form clots. It does not perform all the complex tasks that natural blood does. But as a temporary oxygen bridge during life-threatening emergencies, it holds tremendous promise. The journey toward artificial blood has been long and filled with setbacks. Yet each decade brings better technology, better understanding of biology, and more advanced clinical testing. What once seemed impossible is now scientifically achievable in controlled settings. The question is not whether artificial blood can carry oxygen. We already know it can. The real challenge is making it safe, stable, and effective enough for widespread use. If researchers succeed, the future of medicine could change forever. Blood shortages might become a problem of the past. Emergency care could become faster and more accessible. And millions of lives around the world could be saved simply because oxygen reached tissues in time. Artificial blood may not replace human donation completely, but it could become one of the most important medical innovations of our time.